Hello, everyone, and welcome to this week's edition of the Extracellular Vesicle Club. I'm very happy today to welcome Pasquale D'Acunzo, who's actually in Italy right now, but he's working at the Nathan S. Kline Institute in New York, also affiliated with New York University School of Medicine, and Efrat Levy. So welcome to both of you. I'm really pleased that you've decided to share your work with us today and to take questions from the audience. And Efrat, I think you're somebody that I've really looked up to for many years because you were one of the first people who began to study EVs in the brain. So I'd just like to take a couple of minutes here and ask you a few questions. First of all, I'm wondering if you could share with us how you first became interested in extracellular vesicles. When did you first hear of EVs, and what was it about them that you thought was interesting to study?
So we've been studying Alzheimer's disease and different factors that affect the disease, and starting by looking for mutations in the amyloid precursor protein and studying amyloid beta and processing of the precursor protein. When the first paper came out of showing that neuronal cells secrete extracellular vesicles that contain the amyloid precursor protein and a different metabolic fragments of it and associated with the amyloid beta, I started thinking what is happening actually in the brain. And one of the reasons we are very interested in is that we have been studying the endosomal lysosomal pathway in the brain of Alzheimer's disease patients. I don't know if everybody is aware of it, but actually amyloid deposition is not the earliest pathology, or even tau pathology is not the earliest that was identified in Alzheimer's disease. It is then I think that the early endosome pathology and then late endosomal pathology are the earliest pathologies that can be identified in Alzheimer's disease patients, in Down syndrome. And now we also show it in carriers of the amyloid apolyprotein E4. So we were thinking that it's very possible that there will be an interconnection between endosomal pathology and release of exosomes, specifically exosomes. But we did not want to go in vitro. We thought it will be much better to do it in vivo. And that's why we developed the first method to isolate extracellular vesicles from brains, and then different people did different modification of the method. And finally, Pasquale joined our lab and then did an additional modification, especially because we have seen earlier that the extracellular vesicle contain a lot of mitochondrial proteins, and there is a difference in the content in Down syndrome brain in the amount of mitochondrial protein. So I recruited Pasquale from Rome because of his expertise in mitochondria, and that's where we are. And that led to this finding of specific vesicles that contain mitochondrial proteins.
Well, Efrat, thank you so much for that that perspective. And it's good to hear the background there, and thank you for all of your contributions here. So I'll turn it over then to you and Pasquale to share the screen and to take us through this very exciting recent work.
All right. So good morning or good afternoon to everybody. My name is Pasquale D'Acunzo, and I work in the lab of Professor Efrat Levy at Nathan Kline Institute in New York. And today I'm glad to show to you our newest data on the earliest, our newest paper, in which we describe a novel population of extracellular vesicles of mitochondrial origin that we have isolated from the brain parenchyma, and that we have named the Mitovesicles. I don't have to go too much into details on what EVs are, because the audience knows, but just as a very quick introduction, classically, two main subtypes of EVs have been described, namely microvesicles, which bud from the plasma membrane and are larger EVs of around 100 or 500 nanometers, depending on the nomenclature used. Some people, for bigger vesicles, use the name of oncosomes or even other kind of names. But anyway, microvesicles, originally, they originate from the plasma membrane, while exosomes originate as intraluminal vesicle in multivesicular bodies into the cell. In this sketch, you can see the relationship between endosomal pathway and the exosomal release. So basically, when material is endosized, early endosomes are formed, which later can undergo a process of maturation by which they start invaginate material within the lumen of the endosome. And so through this process, they start producing intraluminal vesicles. Fully mature early endosomes are called late endosomes or multivesicular bodies, which eventually can either fuse with the lysosome for degradation of the cargo or can fuse with the plasma membrane to release intraluminal vesicles as exosomes. The cargo loading of material from the cytosol to the intraluminal vesicles, we all know, is not random, but is mediated by complexes such as the S-core complex, which is found on the membrane of the endosomes. EVs are relevant for neuronal pathophysiology, and this is shown by several papers from our lab, but also from other groups. EVs are relevant for cell-to-cell communication, given that the cytosolic cargo, which is recruited into these intraluminal vesicles, can be, as well as in the microvesicles actually, can be shuttled from one cell to the other, and so is important for this kind of communication. We also have shown a clear and important role of EVs, and more specifically of exosomes, in the disposal of cytotoxic material, such as C-terminal fragments of the APP protein, which is neurotoxic for the cell and accumulates into the endosomes and is released by the cell through exosomes more prominently when a lysosome is blocked, the lysosomal activity is blocked. However, on the other side, the same pathway is responsible for the spreading of this cytotoxic material to neighbor cells. So it's very important in the process of neurodegeneration. EVs are also important for gene expression. We all know that EVs are loaded with microRNA that can alter the messenger RNAs present into the recipient cell. And lastly, they are important in the regulation of immunity. It has been shown that EVs are able to polarize macrophages, but also microglia, either in a pro-inflammatory or anti-inflammatory phenotype. And this is particularly important in neurodegenerative diseases in which neuroinflammation is a hallmark, such as in Down syndrome or Alzheimer's disease. We also all know that EVs can be isolated from plasma, as well from other biofluids. And so by such, they are potential attractive biomarkers and valuable vectors for drug delivery. We have seen with the COVID vaccines that a technique based on liposomes, which are basically artificial EVs, is successful to drive messenger RNAs to recipient cells. Our lab was the first to develop a method to isolate EVs from the brain. In this sketch, you can see a summary of the method we developed, and the method was developed by a colleague of mine, which is Rocio Perez Gonzalez, in 2012. The method relies on a papain treatment of an hemibrain, of murine hemibrain, followed by a series of filtration steps and centrifugation steps to discard and eliminate cells, undigested materials, such as large and small debris. At the end, the pelleted EVs is washed in PBS and loaded on a sucrose density gradient, and is found in fractions that have been named BCD, which have been analyzed by several techniques such as electron microscopy, NTA, Western blotting, mass spec, lipid analysis, and so on, like nucleic acid analysis. This method permits the isolation of clean EV preparations. However, is not suitable for the fractionation of EV subtypes. As you can see, the fraction BCDs do not really separate between subtypes of EVs. However, what we could clearly see is that the distribution of markers of several EV subtypes, such as microvesicles or ectosomes, is not distributed equally. So, for instance, microvesicle markers, such as annexin A2, is mainly found in fraction B, while exosome markers are mainly found in fraction C and D. So starting from these early data, we developed a new method to separate subtypes of EVs. And this method relies on a iodixanol step gradient, which is also a high resolution gradient. So basically, you can use several steps which are very close in their density in order to more finely separate EV subpopulations. With this method, we obtained 8 EV fractions, which were analyzed, as I mentioned earlier. And as a starting point, we analyzed these fractions morphometrically, so using NTA and quantifying the amount of vesicles found in each fraction. The fractions containing more EVs are fraction 1 to 5, and the fraction containing less EVs is fraction 8. Fraction 8 more or less accounts 1% of the total EV population. However, when we analyzed by VCA the protein contents of the fractions, we found that this did not follow exactly what we saw in terms of a number of EVs. For instance, fraction 8 was not differentially, the protein content, in fraction 8 did not differ significantly with fraction 1 and 2, for instance. So we crossed the two data. We quantified the amount of EV per fraction, the amount of protein, per EV per fraction. And what we found is that fraction 1, 2, and 3 were homogeneous and contained a low amount of protein amount per EV, while fraction 8 was the fraction containing EVs with the highest protein amount. Fraction 4 to 6, 7 were somehow in the middle. This data demonstrate a distribution of 3 different types of EV containing a differential amount of protein per EV. We then analyzed by NTA the size distribution of these EVs, and we found that fraction 1, 2, and 3 were similar and contained larger EVs compared to fraction 4, 5, and 6, while fraction 8 was somehow had an intermediate phenotype. Then we analyzed by Western blotting several markers that have been described to be specific of microvesicles and exosomes. Markers for microvesicles such as annexin A1, annexin A2, and annexin 5 were mainly found in fractions 1 to 3, so in low density fractions, consistent with the larger trait that we see by NTA in these EVs. Exosome markers were instead mainly found in fraction 4 to 6. Fraction 7 contained a mixed phenotype, And interestingly, we saw that some EV markers, such as CD63, were high in fraction 7, or at least at the same level of the other exosome-containing fractions. But escort proteins, such as ALIX and TSG101, were very low, if any, in fraction 7, suggesting that the nature of the exosomes found in fraction 7 is different from what found in fraction 4 to 6. Also, what we found is that fraction 8 did not contain any marker of ectosomes or exosome, suggesting that the EVs found in fraction 8 could not be classified as ectosomes or exosomes. Instead, they contained several mitochondrial markers, and more specifically, we found that the markers came from the outer mitochondrial membrane, the inner mitochondrial membrane, and the mitochondrial matrix, suggesting the presence of double membrane EVs in this fraction. We name these newly identified EVs as Mitovesicles. You can also see, as an internal control, we checked for the presence of GM under 30, as well as lamin A or beta-actin, which have been described to be negative in EVs. And as you can see, our preparations were clean and not contaminated by intracellular contaminants. We also checked for the presence of endosomal markers, and this because, as we were mentioning earlier, exosomes derive from endosomes, and consistently we found that endosomal markers are mainly found in fraction 4 to 6. So following what we saw using CD63 and other validated exosomal markers. Also, we saw that fraction 8 again was negative for all these markers, and also fraction 1 to 3 were mainly or negative or very low in these markers. This again reinforced the exosome nature of the vesicles found in fraction 4 to 6. Next, we analyzed the other proteins that have been described to be found in exosomes or in microvesicles. For instance, we checked for the presence of 2 lipid raft proteins. One is caveolin-1, which is a marker of caveolae, and another one is flotilin-1. As you can see, caveolin-1 is mainly found in fraction 1 to 3, suggesting that caveolin and caveolae can be a source of microvesicles. Flotilin instead was mainly found in exosomes, but also in fraction 3, which contained mainly microvesicles, suggesting that fraction 3 may contain a subset of microvesicles with high levels of flotilin-1. LAMP2, which is a protein found in the endolysosomal pathway, peaked in fraction 4, but not in fraction 5. So somehow different from other endosomal proteins analyzed, again suggesting the difference in subpopulations of exosomes and microvesicles in different fractions. Next, we analyzed the presence of lipids and the lipidomic composition of our fractions. Similar to what already described in literature for EVs from cell culture media, we found that cholesterol and not phosphatidylcholine is the main lipid found in microvesicles and exosomes. And again, this is consistent with what reported in literature. This because cholesterol has a different three-dimensional structure and can basically can induce spontaneous membrane curvature when compared to phosphatidylcholine, which is instead mainly found in flat membranes. Interestingly, what we found is that fraction 8 had a lower amount of cholesterol, but higher amount of phosphatidylcholine when compared to other fractions. And this again suggests that the nature of mitovesicles is unique and does not mirror with what is found in earlier fraction. This also is consistent with the mitochondrial nature, given that mitochondria are known to be cholesterol-poor organelles, and so the amount of cholesterol is lower. We also analyzed other types of lipids in our analysis, and what we found is that while galactose and bromide is mainly found in fraction 1, so in microvesicles, PA, phosphatidic acid, which again stimulates a negative curvature, is mainly found in intermediate density fractions. It's particularly low in fraction 8, but fraction 8 is enriched in phosphatidylethanolamine, which also can stimulate a negative curvature. Phosphatidylethanolamine is particularly rich in mitochondria, and so again, this further reinforces a mitochondrial nature of these EVs, also in terms of lipids. Lastly, we analyzed the presence of cardiolipin. This because cardiolipin is a lipid that is almost exclusively found into the cell, into the inner mitochondrial membrane. Consistently with the other data, cardiolipin levels in fraction 1 to 6 were basically negligible, less than 1%, and peaked in fraction 8. In short, what we can say is that lipids found in mitovesicles can confirm the mitochondrial origin of these EVs. This because we found a lower amount of cholesterol, higher levels of phosphatidylcholine or PC, and higher level of phosphatidylethanolamine or PE, and the presence of cardiolipin to around 2 or 3%, which more or less is consistent with what's seen in mitochondria. Then we analyzed our vesicles through EM. In the red insets, you can see a classical transmission electron microscopy of negative stained EVs. And so you can see what we are usually used to see, so basically cup-shaped EVs. We also applied a different type of imaging. We did a cryogenic electron microscopy. So basically through cryo-EM, you can maintain endogenous electron density because these EVs are not stained, but also your shape. So EVs appear more spherical, and fraction 1 to 3 were particularly enriched in EVs with an electron-lucent core, while fraction 4 to 6 had higher levels of a different type of EVs that you can see in the enlargement indicated with B, with a granular core, suggesting that also that microvesicles and at least escort-dependent exosomes can be differentiated by their morphology. We also found that fraction 8 contained a totally different type of EVs, was enriched in EVs with a double membrane and an electron-dense matrix. And this again was consistent with what we see by Western blotting, given that we saw that both protein for the outer and from the inner membrane are present in these EVs. So as conclusions of this first part, we set up a gradient to separate several subtypes of EVs. And using this gradient, we were able to isolate a newly identified, a previously unidentified type of EV of mitochondrial origin, which is negative for all the endosomal, exosomal, and microvesicle markers tested. These EVs contain mostly mitochondrial proteins, and so they have a mitochondrial origin, both in terms of proteins and lipids, and we have named these EVs mitovesicles. Next, we wanted to characterize more deeply in detail the composition of mitovesicles, and also we wondered if we can define mitovesicles as mini mitochondria if they are mitochondrially competent, for instance. But also we wondered what stimulates mitovesicle secretion and how are they secreted. So first of all, we did a mass spectrometry analysis of fraction 8 EVs with positing that if mitovesicles are mini mitochondria, they should mirror what found in intracellular mitochondria. However, this is not the case. intracellular mitochondria, there are several databases that report the mitochondrial proteome, but they all agree more or less in around 1,000 or 1,200. This is the range protein that can be considered intrinsically mitochondrial. Of these proteins, we only found 279 genes proteins, so a small subset of it. So we clustered these proteins according to their function. And what we saw is that the proteome is particularly enriched in proteins involved in energy production and catabolic pathways, while there are other kind of pathways that were scarcely represented or completely absent. For instance, over more than 80 subunits of the mitochondrial ribosome, we did not detect any of them. So again, suggesting the specificity of the cargo found in mitovesicles. We next analyzed by Western blotting. We wanted to confirm the presence or absence of these proteins by Western blotting. And we also wanted to compare the levels of this protein compared to the brain homogenate. So here you can see in this slide that with pH, we indicate the brain homogenate, and we compare the levels of some of the proteins found in our mass spec. And we found that some of them, such as VDAC or MAUA, MAUB, were higher in fractionate when compared to the brain homogenate. On the other side, other proteins were lower in fractionate when compared to the brain homogenate, such as OPA1. And again, this suggests that what we see in fractionate does not mirror exactly, at least in terms of amount of protein, what we see in intracellular mitochondria. Lastly, we checked for the presence of proteins that were negative for in our mass spec to confirm their absence. For instance, here you can see on the right part of this panel that fractionate that the EVs, so mitovesicles, do not contain DRP1, as well as protein abundantly found on intracellular mitochondria, such as TOM20 or mitofusin-2. We also found that when we analyzed the PINK1, we found that only a specific isoform of PINK1 is found in mitovesicles, namely PINK1 delta 2. PINK1 can be found intracellularly mainly as a PINK1 full-length protein, but also as two isoforms which are basically cleaved from the full length. They are called the PINK1 delta 1, which is 53 kilodaltons, and the PINK1 delta 2 that is 44 kilodaltons. As you can see in the brain homogenate, the most prominent isoform is the full length, which is 63 kilodaltons, while in mitovesicles, they are completely negative for the full length and they only contain PINK1 delta 2. So again, this suggests and further reinforce that the mitovesicle proteome is specific when compared to mitochondria. Next, we analyzed, given that mitovesicle proteome was enriched in catabolic proteins, we analyzed if mitovesicles were able to produce ATP. First of all, we checked if native mitovesicles could retain a mitochondrial potential, which is, the condition by which they would be able to produce energy. And so we incubated these EVs with an uncoupler, which is called FCCP, which dissipates potential. And then we also incubated these EVs with the MitoTracker dthread, which is a dye that incorporates more efficiently in a fully energized mitochondria. Sorry. As you can see in the left part of this panel, roughly by NTA, roughly 50% of the vesicles were positive with the dye mitochondrial dthread, which again incorporates mainly in fully energized mitochondria, but this drops down to less than 20%, suggesting that upon incubation with a drug that dissipates their potential, again suggesting that these EVs retain a functional mitochondrial potential at the steady state. Subsequently, we incubated these CVs with several precursors for ATP production, such as ADP, phosphate, and malate, and pyruvate. And we incubated these CVs at 37 degrees in the presence or absence of drugs that block specifically mitochondrial electron transport chain. These drugs are called oligomycin and antimycin and are indicated here with OA. As you can see, after 12 minutes of incubation at 37 degrees, the amount of ATP was roughly doubled in control conditions, suggesting the production of ATP in a short time. However, when we incubated Mitovesicles with oligomycin and antimycin, we did not see any increase, any significant increase in the amount of ATP with time, again suggesting that these CVs are able to produce ATP specifically through the electron transport chain. Lastly, given the presence of MAO-A and MAO-B, which are monoamine oxidase and are involved in neurotransmitter metabolism of monoamines such as dopamine, serotonin, and noradrenaline, we checked the ability of Mitovesicles to retain this monoamine oxidase activity. We incubated these EVs with an analog of monoamines, which is normally quenched. And when it's oxidized, it releases this fluorescent dye. So basically, you can quantify the amount of the enzymatic activity of the monoamine oxidase. As a control, we treated our EVs at the same time with pargilin, which blocks monoamine MAO-A, monoamine oxidase A, chlorgilin, which inhibits MAO-B, and a combination of both. As you can see, when we incubated with both drugs, we did not see any significant, basically, increase in the enzymatic activity, suggesting that the activity we saw is specifically related to the enzymes MAO-A and MAO-B. Then we wondered what stimulates mitovesicle secretion. So we treated the cells in vitro with antimycin A, which, as I mentioned earlier, blocks complex 3, so blocks basically the electron transport chain and stimulates what is called electron leak. So basically what happens when you incubate cells with antimycin A is the production of ROS. So ROS accumulate within mitochondria, but as reported many times by several groups, this does not induce mitophagy because the membrane is not depolarized. So first of all, we checked in the cell lysate if our treatment was able to impair mitochondria, and we analyzed the OPA1 level. So as you can see, upon antimycin A treatment with the Roman letters 1, 2, and 3, you can see independent experiments. So upon antimycin A treatment, OPA1 long isoform level decreases while OPA1 short level stays the same. So when the ratio between the short over the long term goes up, this is a sign of mitochondrial fragmentation. And so this confirmed that our treatment was able to impair mitochondria. In these conditions, we isolated EVs from the cell culture media, and we demonstrated that all the mitochondrial proteins analyzed were higher upon antimycin A treatment compared to the baseline, to the control. Suggesting that mitochondrial dysfunction enhances mitovesicle secretion. In parallel, another group, the Capogiannis group, demonstrated, so this is not our data, demonstrated that mitochondria are able to pinch off vesicle within the cell, and these protrusions and these vesicles are enhanced upon antimycin A treatment. They also show that, as you can see in this image, they also show that these vesicles may contain both inner membrane and outer membrane. And this would suggest that mitochondria, upon dysfunction, or at least upon a dysfunction that does not induce mitophagy, are able to pinch off vesicles. And we also demonstrate that probably these vesicles are also secreted in the extracellular space. Interestingly, they found these vesicles and protrusions in mouse, rat, Drosophila, but even in planarian flatworm neurons. Planarians are the simplest animal with a brain-like structure. It's basically worms. And this would suggest that this mechanism is evolutionary conserved from the most simple animal containing a brain. Again, antimycinase stimulates the production of these vesicles. So in the end, this is the model we developed. We think that mitochondria, we and other groups, as I showed before, think that mitochondria are able to pinch off vesicles containing both the outer and the inner membrane. Probably these vesicles are incorporated somehow into multivesicular bodies. Here you can see this is our data. You can see an image, a photomicrograph of a neuron in the brain of a mouse containing, this is a clear multivesicular body. So you can see several ILVs, intraluminal vesicles, but also, although infrequently, but we saw them, vesicles with a double membrane within multivesicular bodies. So our idea in our model is that somehow mitochondria-derived vesicles can be trafficked to the multivesicular bodies and so released into the extracellular space when multivesicular bodies fuse with the plasma membrane, although we cannot exclude a direct production at the plasma membrane. Next, we analyzed the role of mitovesicle in diseases, and more specifically in Down syndrome. Why Down syndrome? Because Down syndrome is basically recapitulates several features found that are driven by mitochondrial dysfunction. Individuals with Down syndrome have mitochondrial alteration throughout their lifespan, and even in embryonic time, you can see signs of mitochondrial dysfunction. And several traits that Down syndrome individuals carry, such as a premature aging, Down syndrome, or Alzheimer's disease, or heart disease, or autism spectrum disorders, are also linked to mitochondrial dysfunction. So mitochondrial alterations is prominent and very important in these individuals. What we saw is, first of all, we analyzed a mouse model of Down syndrome, which is called TS2. And we analyzed the EVs from TS2 mice compared to litter-mate controls. And we quantified the amount of vesicles through NTA. First of all, we confirmed earlier data from my colleague Rossio, by which we saw that the number of exosomes in Down syndrome murine brains is higher. So here indicated with fractions 4, 5, and 6 combined when compared to controls. And again, this was already published by my colleague. But we also found that the number of vesicles found in fraction 8, so basically mitovesicles, are higher, the amount of these vesicles in the brain of Down syndrome or TS2 mice. This is also confirmed by Western blotting analysis. And we also confirmed this in human brain EV isolated from Down syndrome individuals and deployed aged matched controls, deployed animals here and people indicated here with 2M. Next, we loaded the identical number of EVs as quantified by NTA. And what we found in fraction 8, and what we found is that when we load the same amount of vesicles found in fraction 8, several proteins do not change consistently between Down syndrome and control, but some other proteins, such as UQCRC2, VDAC, and SDHB, were lower. And this, of course, these proteins are mitochondrial proteins, and this suggests that Down syndrome not only impairs the number of mitovesicles, but also their cargo. We also analyzed the presence of mitochondria-specific nucleic acids. We were particularly interested in mitochondrial DNA, and this because mitochondrial DNA has been shown by several groups to be strongly pro-inflammatory when is out of the cell. And neuroinflammation is also a feature of Down syndrome. So what we saw is that equal numbers of mitovesicles from the TS2 mice contain more mitochondrial DNA when compared to controls. And so this would suggest a possible contribution of mitovesicles to the inflammation process. We also analyzed mitochondrial specific messenger RNAs. This because mitochondria contain their own DNA, which encodes for 13 genes, and the messenger RNA of these genes is only found in mitochondria. And what we saw is that all the genes analyzed were lower in TS2 mitovesicles when compared to control, again suggesting a problem in cargo loading of these messenger RNAs. Thus, in conclusion, we show that mitochondrial stress enhances mitovesicle secretion, both in vitro and in vivo. In Down syndrome brains, mitovesicle levels are consistently higher when compared to controls. Because the Down syndrome is characterized by mitochondrial dysfunction, so these data are consistent. We also show that mitovesicle content, both in terms of proteins and nucleic acid, is altered in Down syndrome brains. And then we also show that Down syndrome, there is more mitochondrial DNA that is spread extracellularly in the brain, both because you have more mitovesicles, but also because each mitovesicle contains more mitochondrial DNA. And this possibly contributes to the neuroinflammation mechanism in Down syndrome individuals. So, in short, mitovesicles may have a yet unexplored role in oxidative stress response, neuroinflammation, synaptic regulation, this because we have shown the monoamine oxidase activity, and ultimately in brain homeostasis. I want to thank my lab, and I want to thank, of course, Efrat, that is my mentor and introduced me in the EV world and gave me the opportunity to develop this work. But I also want to thank a lot all the other people working in our lab. I want to thank Rocio that started the project before me, Johan, Lital, Kati, that it's always great to talk with you and to have ideas, and you guys have helped me so much in what I'm doing, and all the other people in our lab, all our collaborators. And I want also to thank the funding agencies that helped us to develop this work. And of course, thank you for the attention.
Well, thanks so much, Pasquale, for presenting this work to us. And it's time now to have a bit of discussion. I am allowing people to unmute themselves, so I will call upon you. So please be ready with your microphone, if possible, to ask your questions as I call on you. But first of all, I wanted to just share some results of this little poll that we did, this flash poll. And we found out that of everybody on the call who answered the poll today, which is about 60 people, we have almost half of people who are working on brain-derived EVs in some way or another. However, we see that only a quarter of people have looked for any mitochondrial contents in EVs. So I'm sure that many of us have gotten some ideas here today from this presentation, and there's probably going to be a lot more work on this coming up. So thanks again for sharing. So I'm going to go to the questions now, and I will start with the first one here. So Ganga has several questions. So Ganga, you can go ahead and unmute yourself and ask your questions. And Pasquale and Efrat, please feel free to jump in at any point.
Yeah, okay. I just want to know whether there was any protein estimation done after isolating the exosomes, and if estimated the protein concentration, I just wanted to know what was the concentration that was used for loading into that gel, and also while doing that Western blot analysis.
So the data I've shown is loaded by volume. So what you see usually is done when you do a density gradient, you load equal volumes. And this is also because we were also interested in, also when we analyzed the Down syndrome brain compared to the deployed controls, we were interested to see also the levels of EVs, not only their content. So the idea is that if you load by microliter, basically, so by volume, if you have more EVs in those volumes, you should also have higher levels of your marker. This would confirm what you see by NTA. right? So most of the data I've shown is but it's what usually people do when you do a density gradient is to load by volume, right? So we did an analysis of the protein concentration, and I showed it in the very beginning in our morphometric data. And what we have seen is that different EVs have a different protein concentration per EV. This is actually consistent with the fact that we saw that in low density fractions, you see a lower amount of protein, per EV. This goes up as the fractions goes up. And this is consistent with the fact that low density fraction basically contain these EVs with the lower amount of protein. This because usually in also when you analyze, for instance, a low density lipoprotein or a high density lipoprotein, what really matters in terms of density of a vesicle is how many proteins they have per EV. So the fact that you see EVs with a higher amount of protein per each EV, and this is found in a higher density fraction, this is something that is consistent. So we were particularly happy about that.
Okay, yeah, okay. One more question. Yeah, I just want to know, you had already told that you have isolated that mitovesicles from that culture media. Okay, I just wanted to know what was the starting volume you used for that isolation.
So we all know that when we isolate EVs from the culture media, the amount of EVs is very low in general. So based on our data, the amount of mitovesicles is like a minor component of the total EV population. So what we did actually was to isolate total EV from the culture media. So we did not isolate only mitovesicles. It's a total EV pool. And then we analyzed by Western blotting, specifically mitochondrial markers. We analyzed several markers. All of the markers were going in the same direction and were strongly up upon antimysin A. And again, consistently with what's in the brain, this suggests that the secretion and also with the literature, I showed that independently from us, other groups have shown that mitochondria are able to produce vesicles, and this production is dependent on the stress you give to the mitochondria. So when you stress the mitochondria, they pinch off more vesicles, they produce more vesicles. So consistently with that, that's what we did. We analyzed upon antimysin A, and we saw that consistently with that, also the secretion of those vesicles, probably because you have more production, is enhanced. Okay. And over the volume, we used basically each experiment is done on the big plates, like 15 centimeters plates, and we use the fibroblast, human untransformed fibroblast, a very low passage number around the P7, P8, around that, and a full plate over 24 hours of treatment. That's more or less the amount of EVs we, in terms of volume.
Yeah, okay. Okay, one more, sir. Have you ever checked on any particular cues or signals that helps this microvesicle to fuse to a particular target or something like that?
Can you repeat your question? Sorry.
Okay. Any particular cues or signals that help this mitovesicle to fuse to a particular target cell or?
So if I can jump in here, if I understand correctly, you're asking, are they targeted to the MVB, for example, or to the plasma membrane, and what might be the signaling?
Yeah.
So we think that for several reasons, we think that mitovesicles are targeted to the multivesicular body. For instance, I showed MAO-A data and MAO-B data. Monoamine oxidase usually into the cell are phased to the cytosol. So the fact that when you incubate your mitovesicles and these activities retained without any saponin or any treatment that disrupts your membrane, right? But the activity, enzymatic activity is retained, meaning that your enzyme faces basically the extracellular phase, right? So it's out of the cell. So basically the topology is retained and is exactly the same of what you see in mitochondria. This also tells you that the outer membrane comes from the mitochondria. So it's unlikely that the outer membrane comes from the plasma membrane, for instance. This is one of the indirect evidence that we have. And also by literature, we are not the first one describing the possible presence of other groups I've described, but also the McBride group. They have shown that multivesicular bodies may contain vesicles coming from mitochondria. What we have done, we have checked in neurons and by EM for the presence of double membrane EVs into the multivesicular bodies of neurons, and we found them. So it's infrequent, and this is also, we think it's also okay with what we get in terms of data, because only 1% of the EVs, it's actually mitovesicles. So the fact that we see this infrequently, it's consistent with that, but we saw that there are double membrane EVs into the multivesicular bodies. The way they reach the multivesicular body is still under investigation, and so we have some ideas, and we are studying it.
Very good. Well, thank you for the questions, Ganga. Let's move on to a few questions now about the contents. Starting with Mike Paulides. Mike, do you want to go ahead and ask you had a couple of questions?
Yes, thanks, Pasquale. Very nice talk. My first question is related to the presence of cardiolipin in your vesicles. It's really compelling evidence that they're of mitochondrial origin. We've tried to analyze using antibodies, which are unreliable, and mass spec requires huge amounts of samples. So I was particularly interested in how you reliably analyzed for cardiolipin in your vesicles.
So the analysis of cardiolipin was done by HPLC, so by chromatography. And this because cardiolipin is a very particular lipid, contains basically 4 tails. Usually we know the phospholipid have 2 tails, right? They have a polar head and 2 tails. Cardiolipin, they contain 4 tails. They are very odd in this sense. So they run completely different by chromatography, in the chromatography gels. So the way we analyzed the levels of cardiolipin was basically to load EVs by volume, as I showed before. And then we quantified using a standard curve with the known amount of cardiolipin to basically have an idea on how many microgram of cardiolipin we got into our fractions. And then we transform it in molar fraction to have the molar ratio. That's the way we did it.
Have you tried using an SS31 peptide labeling at all to label the vesicles with specific for cardiolipin?
We have not. We have not done that. The idea was to analyze the lipidomics as a whole. So we did all by HPLC, and we quantified at the same time several types of lipids. So yeah, we have not done that. It's a very great idea. Thanks.
A second question is totally unrelated. It came up later in your talk, as I wondered if you had actually analyzed for mitochondrial tRNA derived fragments in the mitovesicles from the Down syndrome samples you had.
So we have not. The data we have done was done basically by standard qPCR. So we used for the mitochondrial DNA, we used the pre-treatment with the RNAs to eliminate all the RNA present. So we were sure we were only quantifying with the same primers. This is because, when mitochondrial DNA do not contain introns, so you cannot have primers that discriminate between them. So the way we analyze this nucleic acid was for the mitochondrial DNA, we pre-treated the isolated nucleic acids with RNAs. while for the messenger RNA, we used basically the same primers, but pre-treating with DNAs and keeping the RNA. So that's the way we did it. Given that we did not perform a RNA-seq or any other kind of high throughput data, we only did the qPCR. Yeah, we did not analyze the tRNA, but only the messenger RNAs.
Okay, thank you very much.
We next have a question from Paula Laredo. So Paula, feel free to unmute yourself, but I'm just going to ask this question in the interest of time. Do reactive oxygen species levels in the mitovesicles correlate with those in the mitochondria in the cell? And Paula, you can elaborate if you'd like.
Okay. Hi, can you hear me?
Yes.
Okay. Hi, Pasquale. I wanted to be just, you mentioned in your data that the ROS levels changes or induce the budding of the mitovesicles. So I was just wondering if this ROS levels inside changes also, and subsequently, if there are changes in the lipids, like peroxidation or any ROS changes related to the lipids.
So we tried. So we tried to quantify ROS levels using mitosocs, but the problem is that the amount of mitovesicles that you get from a brain is very low compared to the what you get from the brain of mitochondria, basically. So we never reached any signal over the background in any condition. So basically the amount of mitosocs incorporated into the mitovesicles is so low that the machine was not able to measure it. So we do not know. It's very likely, I think, but we don't have data to demonstrate that ROS levels change in the mitovesicle from Down syndrome brain or not. But I think it's likely, but again, we don't have data for that. I tried, but it didn't work. The same applies for the peroxidation. We tried using an antibody by Western blotting for 4-HNE, which is a peroxidized lipid, which is linked to proteins when peroxidation goes on, but the level were basically undetectable in fractionate in any condition. So I don't have an answer in this sense, but I think it's likely.
Okay, and just out of curiosity, any changes on oxidation or reduction of the proteins? If you checked if they change compared to the mitochondrial to the progenitor cell?
We did not check for that. So for sure, the fact that the potential is retained and the fact that ATP is produced would suggest that you have a similar system to for the redox, right, that are typical of the electron transport chain, and also to recover from that, right? So it would be likely, but I have not checked it specifically for that.
Thank you.
So we have a couple of questions here, and I'm just going to kind of summarize them about whether you think that mitovesicle release is going to be different from different cells, including, from different cells in the brain in Down syndrome. So this is from Seema Singh and also from Susan Goals. So it's a great question, and it's something we are testing to date. So what I can tell you is that it was very helpful for us, the fact that the paper was published in 2019 on Nature Neuroscience. They show that mitochondrial traits from astrocytes are completely different from the mitochondrial protein found in neurons, and they find several proteins that are uniquely found in neuronal mitochondria versus astrocytic mitochondria. We checked several of them, and we found both of them. So, for instance, MAO-B is exclusively found in astrocytes, such as sideroflexin 5, while OCHA2 and other mitochondrial proteins, MCU, and so on, are only found in neuronal mitochondria. What we have seen is that both of them are found in fractionate. We are now trying to set up a protocol to see if we can
somehow sort or differentiate between them, and so keep going with this differentiation. By a metabolic point of view, neurons and astrocytes are totally different. Astrocytes relies mostly on oxidative phosphorylation, and neurons relies more on glycolysis, but mitochondria are more specialized in other kind of things. For instance, calcium buffering. So you have mitochondria that are found in the synapse that are basically full of calcium, and they buffer the calcium that goes inside. And so there is somehow although we always think about mitochondria as just the powerhouse of the cell, actually what we know in the brain, there is a very specific differentiation and heterogeneity in terms of mitochondria and within the same cell in different zones. So it's hard to answer to this question in terms of specificity, but of course I can tell you that both cell types release a lot of mitovesicles, probably astrocytes more, but we're still working on it.
Thanks for the question. So we're going to have to wrap up soon, but I see there's still some very interesting questions here. I will send over the chat record, Pasquale and Efrat, so that if you have any follow-ups to any of these, but there's one here from Michael Spanos who asks if these might just be semi-functional mitochondria.
Okay, so it depends what functional mitochondria means, in the sense that mitochondria have several functionality, right? They produce energy, but they also synthesize proteins, right? They have ribosomes and they synthesize proteins. They buffer calcium. They regulate, by the metabolic point of view, several neurotransmitters, right? So you have several functions that mitochondria have. According to our mass spec, some of these functions are retained, and we have data showing that, for instance, ATP is produced, but some other functions are not there. We do not see, for instance, the polymerase gamma that is responsible for DNA, mitochondrial DNA replication. We did not see, both by mass spec and by Western blotting, we did not see any protein related to the MAMS, for instance, the mitochondria-associated membranes with the endoplasmic reticulum. For instance, we checked for TSPO, which is a classical cholesterol carrier found in those MAMS and responsible for the uptake of cholesterol into the mitochondria. We mentioned mitochondria are not able to produce cholesterol, so they need to import it from the endoplasmic reticulum. We did not find it. Other like messenger RNA production or translation is not found there. So several functions are not there at all. We do not see any proteins involved in those functions, but some other are. So what we think is that these are units that are capable for doing some activity, but not others. That's our conclusion of what we get from our data.
So hello, it's Michael. So yeah. My main question is, how confidently can you characterize those vesicles in terms of markers or a group of markers? Let's say if I want to isolate those vesicles, would you advise that they should do it as you did it via the protein content, the size, or are there any specific markers? Because I've seen your paper, and congratulations. And there is no markers for these or microvesicles on those, right? So is there a marker, because there are markers of exclusion then, but is there a marker of inclusion? Is there a specific markers for those microvesicles?
So we have not markers, but I think this is a problem which is common also for other EVs, in the sense that when you see, let's say, CD63 is a marker of endosomes also, right? I mean, a specific marker of exosomes, I don't know what it means, in a sense that, you know, like every protein you see in exosomes, what?
I understand, yeah. I see your point. Yep. One last question, and thank you so much for this. You talked about neuronal and astrocytic specificity, and that is amazing. If this can be actually achieved, have you checked any other organs releasing mitovesicles?
so we have not, but the data we have produced in vitro are from fibroblasts. So we know that fibroblasts secrete mitovesicles. Also, that was a question of a reviewer of our paper, and stimulates us to check in databases for plasma, right? So you have several databases of proteomics in plasma, and we checked that. They are available online, like Protein Atlas. There are several, freely available software, so everybody can check. And we were amazed by the fact that not only in the plasma, by mass spec, you can see mitochondrial proteins, but they match exactly what we see in the mitovesicles, in the sense that you see proteins involved in catabolic pathways, in ATP production, and blah, 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 but we do not see several markers that we have not seen in our mitovesicle data. So what we think is that also in plasma, you can find them. Now, we are trying to establish a protocol to isolate mitovesicle from plasma, and I'm working on that. We know all the problems that this encompass. Also consider that mitovesicles are very low compared in terms of number compared to other EVs. So it's particularly struggling, but we are trying to do that.
Okay, thank you so much.
Of course. Thank you.
All right, well, we will have to conclude our session here. We do have some very interesting questions remaining about the relationship between stress and mitovesicles and apoptosis and mitovesicles. And I'm going to share these with you again as a text file. But thank you all for joining today, for all of your insightful questions, and special thanks to Pasquale and Efrat for joining us and for sharing this very exciting work. So thank you all, and I look forward to seeing you all again very soon. Take care, everyone.
Thank you.
Thank you. Bye.
Bye.